Okay, let's unpack this. You know that feeling. You've got a mountain of information on a really complex topic, and you need to get to the heart of it fast.
That's exactly what we aim to do here.
Today, we're doing a deep dive into a pretty fascinating area, medical foods, specifically those made from natural sources.
And our guide for this journey is the research presented in Medical Foods from Natural Sources by Meera Kaur. It's quite a detailed look.
Right. So our mission today is to really dig into this material. We want to understand, what exactly are medical foods?
Uh-huh.
Why is exploring natural ingredients for them, as this research does, so important? How are they actually developed and tested?
And crucially, what did the research, especially the clinical trials mentioned in the book, actually find? How effective are they?
Exactly. And why should you care? Well, because nutrition is absolutely critical in patient care.
It really is. And how we deliver that nutrition, especially when someone can't eat normally, can make a huge difference. This research explores a different, maybe better way using natural stuff.
Okay, let's start right there. When this research talks about medical foods, what are we actually discussing? How do they differ from, say, regular food or even a vitamin pill?
That's the key starting point. The research makes it clear. Medical foods are their own category. They're formulated for the specific dietary management of a disease or condition.
So not just general health, but something very specific.
Precisely. And there have to be distinctive nutritional requirements for that condition based on, solid scientific principles and medical evaluation. They're used under medical supervision.
Okay, targeted and supervised. That makes sense.
And because of that, they fall under slightly different regulatory rules than conventional foods or supplements, like under the Nutrition Labeling and Education Act. They don't need the standard health claims, for example.
But they still need labels, right? You need to know what's in them.
Oh, absolutely. The research points out clear labeling is still required. You need the product identity, net quantity, manufacturer info, and a full ingredient list.
Order of amount, I assume.
Yes. Descending order of predominance by weight. And all that mandatory info has to be prominent, easy to spot, and in English, though you can add other languages too.
And manufacturing. That can't just be made anywhere, surely.
Definitely not. The research mentions they have to comply with standard food manufacturing requirements, think good manufacturing practices, facility registration.
Right.
And if it's a liquid food in a can, specific rules for low-acid canned foods apply. Plus, and this is critical, all ingredients must either be approved food additives or generally recognized as safe GRS.
What about food allergies? That's always a big concern.
A very important one. The book notes the Food Allergen Labeling and Consumer Protection Act, FLCA. Since 2006, labels must clearly list the major food allergens.
Milk, eggs, nuts, the usual suspect.
Yep. Milk, eggs, fish, shellfish, tree nuts, wheat, peanuts, and soybeans all have to be declared.
Okay, so specialized regulated foods for specific needs. This connects directly to enteral nutrition, or EN, which is a huge part of this research. What exactly is EN?
Simply put, enteral nutrition is giving nutritional support directly into the gut, the gastrointestinal tract, using a tube.
Tube feeding, basically.
Exactly. It bypasses the mouth and throat for when someone can't swallow safely or maybe can't digest normally.
And the research talks about different ways this is done.
It does. It mentions various routes. Tubes can go via the nose down to the stomach, that's nasogastric, or further into the intestine, nasoduodenal or nasojejunal.
Or directly into the stomach.
Right, through the abdominal wall, that's a gastrostomy, or into the jejunum, the small intestine, a jejunostomy, and different feeding methods, too.
Like how the food is given.
Yeah, like giving a larger amount quickly, that's bolus feeding. Or slow, steady, drip, continuous feeding, or feeding at set times, intermittent feeding.
And who needs this kind of support? Is it common?
It's more common than you might think. The book highlights that a significant percentage of hospitalized patients, maybe 25 to 50%, could be malnourished and benefit from it.
Wow, that's a lot.
It is, and it lists specific groups. ICU patients, people with cancer, comatose patients, those with sepsis, difficulty swallowing dysphagia or severe appetite loss, anorexia.
Also gut issues.
Yes, severe diarrhea, problems absorbing nutrients, also children with certain heart conditions, elderly patients, people with severe burns, kidney or liver dysfunction, lung diseases, AIDS patients. It's a broad range.
That really drives home why effective EN is so vital. The research touches on the history of it too.
Just briefly, it mentions some early studies back in the 1950s using sort of basic protein and cereal mixes through early types of tubes. Then, from the late 60s, more complex, chemically defined diets became possible as tube placement techniques got better.
Okay, so we get the basics of medical foods and EN. Now, a key point in this research is about the problems with many existing enteral formulas, the ones based on defined or synthetic ingredients. What issues does the book raise?
Well, it points out that while these conventional formulas do provide nutrients, they can have drawbacks. Cost is one that can be expensive. Palatability can be an issue if someone can taste them. Even though they're mostly for tubes, they often need artificial flavors. But a really key point the research emphasizes is they might lack beneficial natural compounds.
What kind of compounds?
Things like natural stimulants or protective substances that you'd naturally find in whole foods. Things that aren't just basic proteins, fats, and carbs.
So you lose something when you break food down into just its chemical parts.
That seems to be the perspective explored here. And this leads directly into the book's main contribution, looking at developing enteral foods that are potentially more cost-effective and use natural ingredients. Things like barley, rice, mung beans, eggs, milk.
A food-like approach instead of a drug-like approach, as the book puts it.
Exactly. Trying to use the synergy of whole foods rather than just assembling isolated chemicals.
That sounds good intuitively, using real food. But wait, tube feeding. How do you make a liquid from grains and beans that's nutritious and flows smoothly through a tiny tube? That sounds really challenging.
Oh, it absolutely is. The research acknowledges this is a big hurdle for scientists. Getting that high nutrient density from natural stuff into a liquid form that won't clog a narrow feeding tube is, well, that's tricky.
So what's the proposed solution in this research? How did they tackle that?
A major part of their approach was using malted cereals and grain legumes as the base ingredients.
Malting, like with beer.
Kind of, yeah. The process is similar in principle. Malting involves controlled sprouting or germination, followed by drying. It fundamentally changes the grain.
Okay, so that brings us to the specific ingredients and processes they used.
Yeah.
What were the main natural ingredients?
The core ingredients mentioned were barley, rice, mung bean, fresh hen's eggs, milk, and also grain amaranth, the white seed kind.
Where do they get these?
Sourced locally, it seems, poultry farms, local markets. They also use some supplemental things. Spray-dried gum acacia, likely for texture and stability, and standard USP or BP grade vitamins and minerals, plus some extra nutrients like taurine and carnitine to make sure the formulas were nutritionally complete.
Got it. Now, the processing, you said malting was key. Can you break down what that actually involves, according to the book?
Sure. Malting starts with germination, letting the greens sprout under controlled conditions. Then you dry it, which is called kilning. The magic really happens during germination.
What kind of magic?
Enzyme magic. The grain activates or creates a whole bunch of enzymes. Amylases are a big one. They start breaking down the complex starches.
Into smaller sugars.
Exactly. Alpha amylase kind of chops randomly. Beta amylase nibbles off sugars like maltose from the ends. There are also debranching enzymes for specific starch structures, and the research mentions pentosinase as they break down cell walls, which lets the amylases get to the starch more easily.
Okay, so it makes starch easier to digest. Anything else?
Lipase activity also increases. Enzymes that break down fats, not a huge factor for cereals maybe, but relevant.
And then kilning, the drying part.
Right. Drying with warm air. It reduces moisture way down, stops the enzymes, cures the malt, dries the little sprouts. The trick is getting the temperature right, high enough to dry and stabilize, but not so high you destroy the enzymes or nutrients.
So that it affects flavor.
It can. The research mentions small-scale malting often uses contact heat, which can cause slight browning and develops that characteristic malty aroma that's the Maillard reaction between sugars and amino acids. Overall, malting drastically improves digestibility and reduces the thickness, the viscosity, when mixed with water.
Makes sense. Less thick, more digestible. What about other methods? Popping was mentioned for amaranth.
Yes, similar to popping popcorn. They describe adding a bit of moisture to the amaranth grain, letting it sit or temper, then heating it quickly in a hot pan with constant stirring.
And the benefits of popping here.
Several things. It really improves the flavor and smell. It helps kill surface bacteria, which is good for safety. And importantly, it improves starch digestibility too.
Any downsides? Nutrient loss.
The research notes that very high heat can damage some amino acids, like lysine, but it also mentions that improved methods, like using fluid bed systems, can minimize that loss while still getting the benefits.
They also mentioned toasting.
Yeah, a simple dry heat method. Toasting grains or legumes on a stovetop or in an oven. Again, helps kill surface microbes, adds flavor, reduces moisture, which improves shelf life,
Uh-huh.
and it can reduce certain enzyme activities that cause fats to go rancid, useful for things like soybean flour.
Okay, and the last one was spray drying. Sounds more industrial.
It is very common for making powders from liquids, think powdered milk or instant coffee. You typically concentrate the liquid food first, then spray it as a very fine mist into a chamber with hot air.
And the water just evaporates.
Instantly, pretty much, leaving tiny dry particles behind. It's vital to collect these particles aseptically and pack them in airtight containers to keep them dry and safe.
Anything crucial to watch during the process?
The temperature of the air coming out of the dryer is a key indicator. It reflects quality and process efficiency. If it suggests heat damage might be happening, the research says you can adjust the feed rate or the inlet air temperature or make the spray droplets smaller so they dry even faster. It's effective, but it does add cost, and you have to be careful about potential damage to sensitive things like proteins.
So using these natural ingredients and these processing methods, the research team formulated 3 specific enteral foods. What were they called again?
Right, there were 3 prototypes. GEFLC, that's general category low cost, then GEFRT, general category ready to eat, and finally DEFBP, which was disease specific, designed for burn patients.
How are the ready-to-eat and the burn formula different from the low-cost one and how they were made?
The GEFLC was basically a blend of the processed ingredients, malted grains, milk powder, oil, vitamins, etc. It needed cooking before use. The other 2, GEFRT and DEFBP, were prepared using spray drying, so they are powders ready to be mixed with water, no cooking needed.
Okay, so they made these formulas, then they had to test them, right? What did they find about the basic nutrition, protein, fat, calories?
Looking at the powders, the general ones, GEFLC and RT, were about 16% protein, 12% fat. The burn formula, DEFBP, was higher, around 21% protein, 19% fat, which makes sense for burns.
And when mixed up for feeding?
At a typical feeding consistency, say 25-30% solids, they provided a good energy density, roughly 1,100 to 1,600 kilcal per liter, plus 40-65 grams of protein and a decent amount of fiber, 12-13 grams per liter.
What about those physical properties, like the viscosity we talked about? Did they measure that?
We did. They measured viscosity using a Brookfield viscometer, gives you a number in centipoise CP, showing how thick it is. They also looked at solubility, how well it dissolved, and swelling power, how much water it absorbed.
And the key finding
A
there.
really important one. The viscosity of their foods was considerably lower than commercial formulas when compared at the same concentration.
Which is good for tube feeding.
Exactly. Lower viscosity means it flows much more easily through those narrow feeding tubes, less risk of clogging.
Did they test the actual flow?
Yes, they confirmed that even at fairly high concentrations, 20-35% solids, the formulas flowed smoothly under gravity through standard feeding tubes, sizes 6 to 18 French gauge.
Good. They also looked at osmolality, right? How concentrated the liquid is.
Yeah.
The load on the kidneys.
Right, osmolality and renal solute load. The developed foods ranged from about 246 to 500 mosmol per kilogram or liter. They compared this to some commercial examples like Jevity at 300 or Pulmicare at 475.
So in a similar ballpark.
Pretty much in a reasonable range. The research does note that formulas with higher osmolality might need a short adaptation period for the gut just to avoid any potential upset.
Makes sense. What about the carbohydrates? You mentioned malting breaks them down. Did they confirm that?
They did, using a technique called gel permeation chromatography. It separates molecules by size. They found the carbs were indeed pre-digested, mostly smaller chains like maltodextrins.
How did they know it was the malting?
They compared the malted formulas to blends made with unmalted ingredients. The unmalted ones had lots of big, high molecular weight starch peaks. The malted ones had much smaller peaks. It clearly showed the malting process worked to break down the starch.
Okay, carbs pre-digested. What about the protein? Quality, structure.
They checked the essential amino acid profile, found it was well balanced, which is crucial. They also used SDS page, a technique to look at protein size. It showed the proteins were largely intact.
Any changes from processing?
They did note a slight reduction in lysine, an essential amino acid, in the spray-dried formulas, GEFRT and DEFBP. That can happen with heat.
And wasn't there something unusual about the egg protein in the burn formula?
Yes. This was highlighted as quite interesting. Normally, egg protein thickens or coagulates when you heat it, right?
Right.
But in the DEFBP formula, even after heating, it stayed remarkably free-flowing. The research suggests something in the mix, maybe the malt enzymes interacting with the egg, altered its usual coagulation behavior.
That is interesting. And the fats. What kind of fats were in these formulas?
They analyzed the fatty acid profiles. About 65% of the fats were unsaturated, generally seen as healthier. Linoleic acid was dominant in the general formulas, while oleic acid was higher in the Burn formula.
Did the Burn formula have omega-3s?
Yes, DEFBP contained about 5.9% omega-3 polyunsaturated fatty acids, including EPA and DHA, likely from added fish oil, which is often recommended for burn patients. It also had higher cholesterol and phospholipids because of the egg content.
Any thoughts on fat stability?
They raised an interesting possibility that ingredients like the gum acacia and the popped amaranth might actually help protect the fats from breaking down or developing off flavors during storage. But they noted this needed more investigation.
Okay, so the composition looks good. The research also used animal studies to check protein quality more formally, right?
Yes. Standard rat feeding trials, they measured things like PER, protein efficiency ratio, NPR and net protein ratio, RNPR, relative net protein ratio. These all gauge how well the protein supports growth.
And digestibility.
They also did nitrogen balance studies to find the true digestibility, TD, biological value, BV, how well the absorbed protein is used, and net protein utilization, NPU, which combines both. Plus, they checked the rat's organs for safety.
And the results of these protein tests?
Very positive, according to the book. The foods showed good growth-promoting quality based on PER values. They had high biological value, BV, between 85 and 94%, and high digestibility, TD, around 87-90%. This suggests the amino acid profile was well-balanced and usable by the body. And safety. The organ analysis showed everything was normal, indicating the foods were free from toxins. Interestingly, the Burn formula, DEFBP, showed significantly better growth promotion, higher NPR compared to the general formulas, and was pretty much on par with skimmed milk powder, a high-quality reference protein.
Okay, so lab and animal studies suggest these foods are nutritious, safe, and have practical advantages like flow. What about shelf stability and safety for human use?
Microbiological safety was tested thoroughly. Total plate count, coliforms, yeast, mold, spores all met safety standards. They also tested for pesticide residues using standard methods.
Good. And shelf life. How long did they last?
They did storage studies, both at room temperature and under accelerated conditions. They tracked moisture, free fatty acids, peroxide value, both signs of fat spoilage, and pH.
Did packaging matter?
Yes, significantly. Fat breakdown, higher FFA, was faster in samples packed in basic polypropylene bags compared to better barriers like metallized polyester or aluminum foil laminates. The spray-dried powders tended to pick up moisture in some pouches but stayed dry in the foil ones. A slight drop in pH over time was also noted.
And did they still seem okay to eat? Sensory tests?
They did sensory evaluation using scorecards looking at appearance, color, flavor, taste, consistency, overall acceptability. GEFLC was acceptable for about 45 days at room temp, GEFRT up to 120 days. DEFBP, the Burn formula, was still acceptable when its longer study ended.
Any changes in appearance?
The powders tended to brown slightly on storage, more so in the less protective packaging. And remember those interesting stability points we mentioned?
The egg not browning in DEFBP.
Exactly. Despite having egg sugars, which usually cause browning in egg powder unless removed, this formula didn't brown much, again suggesting something unique about the formulation. They also reiterated the idea that gum acacia and popped amaranth might be encapsulating fats, preventing flavor reversion.
And the probiotics.
Yes, they found a surprisingly good survival rate, 60-70% of the lactic acid bacteria through spray drying and storage, possibly protected by the gum acacia matrix.
Okay, lab tests look promising. Stability seems decent with good packaging, but the real proof is in using them with people, right? What about the clinical trials?
Absolutely. They conducted trials after getting ethical approval and patient consent. Two main sets of trials, one using the Burn-specific formula, DEFBP, with burn patients, and another using the general formulas, GEFLC and GEFRT, with general medicine and surgery patients.
Let's start with the burns. Why is nutrition so absolutely critical there?
The research underscores that severe burns cause immense metabolic stress. Patients desperately need support for fluid balance, to minimize muscle breakdown, and crucially, to boost their immune system to fight infection, which is a huge risk.
Makes sense. So what did the DFBP trial involve?
They did initial tests for tolerance and acceptability, which went well. Then a controlled clinical trial comparing DEFBP fed via tube against the standard hospital diet in the control group. A key part was starting the feeding early. within 24 hours of admission if possible.
And what did they measure to see if DFBP made a difference?
A lot of things. Burn characteristics, of course, nutritional status, weight, body measurements, urinary protein loss, blood tests, blood counts, liver enzymes like alkaline phosphatase and SGPT, electrolytes, immune status, both antibody levels and cell-mediated immunity.
Anything else?
Yes, also graft success, GI tolerance, any vomiting, diarrhea, gut slowdown, lactose issues, and the rate of stress ulcers.
That's comprehensive. So what were the key results for the burn patients on DEFBP?
Some really interesting findings. Survival probability was slightly higher in the DEFBP group. Urinary protein loss was significantly lower, suggesting faster kidney recovery.
What about blood tests?
An improved blood picture, better white and red cell counts, and notably a significant increase in lymphocytes, those key immune cells, in the DEFBP group, suggesting better immune function.
And the liver stress.
Liver enzymes initially spiked in both groups as expected, but by the end of the study, alkaline phosphatase and SGPT had decreased significantly more in the experimental group, indicating faster liver function recovery. Cell-mediated immunity was also found to be better in the DFBP group.
And how did their guts handle it? Tolerance.
This was a big positive. They reported no major GI problems, no vomiting, diarrhea, or ileus in any patients on either diet, no lactose intolerance either, which they thought might be due to beneficial bacteria in DEFBP and yogurt in the control diet.
And stress ulcers.
Also notable, stress ulcers occurred in the control group, but not in the group receiving DEFBP. So the research discusses these findings, emphasizing the benefit of early feeding and suggesting the specific components of DEFBP, like the fish oil fats, the branched-chain amino acids. Glutamine likely played roles in the improved immune status and liver recovery.
Okay, really promising results for the burn formula. Now, what about the general category foods, GEFLC and GEFRT, used in that broader group of patients?
Right. These were used with about 100 general medicine and surgery patients, either as their total nutrition or as a supplement.
And the overall outcome there?
Generally positive, according to the research. They reported improved nitrogen balance, suggesting better protein use, improved blood markers, reduced hospital stays, and faster healing of surgical fistulas.
The book also shared some specific patient stories, case studies.
It did, and they really help illustrate the potential impact. One was a straightforward post-op patient where GEFLC worked well as complete or supplemental nutrition.
Was there a case showing better tolerance?
Yes, case 2 was powerful. An 8-year-old boy with a serious abdominal issue couldn't tolerate a standard commercial formula, got severe diarrhea. But when they switched him to the GEFLC tube feed, he tolerated it immediately. His digestion normalized quickly, his health improved, and he was discharged within a month using only GEFLC.
Wow, that's quite a difference. What about case 3?
That was a 42-year-old man, very ill with chronic liver disease, hepatic coma, severely malnourished. He received GEFRT via tube for 45 days, tolerated it perfectly, showed significant improvements in weight, muscle measurements, and his liver and nutritional blood markers all normalized by discharge.
What do they attribute that success to?
The research points to the vegetable proteins, the specific amino acid profile, more BCAAs, fewer AAAs, good for liver disease, the probiotics, fiber, and added nutrients like taurine and carnitine. They specifically discuss benefits of vegetable protein and BCAAs for liver patients.
And the last case?
Case 4, a patient with esophageal cancer, severe swallowing problems, very malnourished, received GEFRT via NG tube. Again, tolerated it without any issues like nausea or fluid overload. After 40 days, showed significant gains in weight and nutritional status indicators, arm measurements, blood proteins. All other functions remain normal.
And the reason for success there?
Linked to GEFRT's good osmolality, fiber, probiotics, and the easily digestible nutrients from malting. The conclusion was it provided excellent support in a very difficult situation.
These stories, along with the broader data, really seem to build a strong case. So the overall takeaway for these general patient foods?
The conclusion presented is that these malted grain and milk-based foods, fortified correctly, can provide optimal, affordable nutritional support for many general patients. And they particularly highlight the low-cost version, GEFLC, as a really valuable option, especially in resource-limited settings like developing countries.
Okay, let's pull this all together then. What are the major conclusions, the big takeaways from this deep dive into Meera Kaur's research on natural medical foods?
I think the main message is pretty clear. Using natural ingredients isn't just feasible, it's actually a very promising way to create medical foods. The research suggests these can be nutritionally balanced, cost-effective, and importantly, very well tolerated by patients.
Better tolerated even than some standard formulas based on those trials.
Yes, that seemed to be a key finding. Processes like malting are shown to be really effective for creating suitable bases. The lab work holds up, and the clinical results look good compared to standard approaches. Probiotics seem to add real value for tolerance, especially with milk ingredients. And the cost factor is a potentially huge advantage.
What does this imply for scientists, doctors, dietitians?
It really points towards a need, or at least an opportunity, for more enteral foods that are maybe more physiologically normal and definitely more cost-effective. Natural sources seem to offer a great pathway, using the inherent benefits of whole food components, not just isolated nutrients.
Did the research suggest where to go next? Future directions?
It did. Firstly, develop more disease-specific formulas from natural ingredients because nutritional needs vary so much. They tackled burns, but there are many other conditions.
And the format? They were powders.
Right. Developing stable, ready-to-use liquid forms was highlighted as a key next step. The current powder form was partly due to stability issues with added nutrients like glutamine in liquid. And finally, they circled back to that really intriguing point about the egg protein in the burn formula.
The non-coagulating egg.
Exactly. They stressed the need for future research to figure out precisely why that happened. What was the interaction between the malt enzymes, the fiber, the probiotics, the fresh egg itself? Understanding that could open up new ways to make stable liquid formulas from natural proteins.
That mystery about the egg protein is a great place to kind of wrap things up. We've really unpacked how this research meticulously explored using natural ingredients and traditional methods for medical foods.
Yeah, building the case with careful lab work, animal studies, and then those compelling clinical results.
So hopefully you've gained some real insight into what goes into these specialized foods, the science behind using things like malted grains or even eggs in this way, as presented in Kaur's work, and the positive outcomes seen in patients.
It challenges the conventional defined ingredient approach in some interesting ways.
Definitely. And as a final thought to leave you with, building on this research, how might those complex interactions between all the components in natural foods offer benefits that we just can't easily replicate by mixing isolated chemicals? And what other natural ingredients or maybe traditional food preparations from different cultures might hold untapped potential for medical nutrition? Something to think about.